As we discussed nucleotide chemistry and the structure of DNA, now we'll discuss RNA, which is ribonucleic acid. Now let us look at the structure of RNA. Unlike DNA, RNA is a single stranded structure with a sugar phosphate backbone, nitrogenous base, and purines as adenine and guanine, whereas pyrimidine is uracil and cytosine, and there is no thymine present in the RNA structure. RNA is a polyribonucleotide. As I stated, it is a single stranded polynucleotide. Phosphodiester bond links the nucleotide. Now look at the phosphodiester bond that links the nucleotide, that is fifth carbon atom of the pentose sugar to the third carbon atom of the another pentose sugar. It is labeled as 5' 3' phosphodiester bond. And formed between 3-hydroxy group of one pentose sugar and 5-hydroxy group of the other pentose sugar of another ribonucleotide. Nucleotides which are found in RNA are AMP, GMP, CMP, UMP. Thymine bases are absent in RNA. Minor methylated thymine and dihydrouracil can be present in the RNA structure. Unlike DNA, Chargaff rule is not obeyed in RNA. This is because of the single-stranded nature of the RNA, and there is no specific relation between purines and pyrimidine contained. During alkali hydrolysis of the RNA, alkali can hydrolyze RNA to 2', 3' cyclic diesters, and this is due to the presence of hydroxy group at the 2' position of the pentose sugar. In Orsinol color reaction, RNA can be identified, and this is due to the presence of ribose sugar present in the RNA. Now, what are the types of RNA which are present in our body? One is mRNA, which is present in approximately 5 to 10% of the total RNA. Transfer RNA, which is 10 to 20%. Ribosomal RNA, which is 50 to 80%, and there are other kind of RNA which are also present in our body. Now look at the cellular RNA and their functions. First is messenger RNA. What it does, it transfers the genetic information to synthesize protein. Heterogeneous nuclear RNA, which is abbreviated as hnRNA, it is a precursor for mRNA and other RNA. During the process of transcription, DNA first synthesizes hnRNA, which gets further processed to form mRNA. The other form of RNA is tRNA, which transfers amino acid to the growing mRNA for protein biosynthesis. Cut it. Transfer RNA, which transfers amino acid to mRNA for protein biosynthesis. Ribosomal RNA provides structural framework for the ribosomes. snRNA, which is small nuclear RNA, it is involved in the processing of the mRNA. Small nuclear RNA is also involved in the processing of ribosomal RNA molecule. scRNA, which is small cytoplasmic RNA, it does the selection of protein for export. Transfer messenger RNA, that is labeled as tmRNA, and it is different from tRNA, which is transfer RNA. It is not present in the eukaryotes, it's present in prokaryotes, and it adds short peptide tags to protein to facilitate degradation of incorrectly synthesized proteins. So this is about the various kind of cellular RNA and their different functions. Now we'll discuss ribosomal RNA. As the name suggests, ribosomal RNA, it is found in ribosome and it is abbreviated as rRNA. Eukaryotic ribosomes are factories of protein biosynthesis, and it is composed of two major nucleoprotein complexes, that is 60S and 40S subunit. 60S subunit contains 28S rRNA, 5S rRNA, and 5.8S rRNA. 40S subunit of the ribosomal RNA contains 18S rRNA, and the main function of ribosomal RNA is for protein biosynthesis. Now let us talk about messenger RNA. Messenger RNA, during the transcription process, it is that DNA to RNA is called as transcription. And during this process, DNA first synthesizes hnRNA, which gets further processed to form mRNA. And this mRNA is involved in protein biosynthesis. Now we'll talk about the structure of hnRNA. There's a capping at the 5' end, and there's a capping at the 5' end, and it has 5-methylguanosine. This 5-methylguanosine prevents hydrolysis by 5' exonucleases, and it is also the recognition site of mRNA for protein biosynthesis. Okay, so this is about the 5' end of the mRNA. Now there is a 3' end which has poly A tail, and the 3' end contains polymer of adenylate residues, which is about 20 to 250 nucleotides. This end provides stability and prevents the RNA from the attack of 3' exonucleases. Now, since we know that this RNA is involved in the protein biosynthesis, so this RNA has codon region, and different codons code for different amino acids. Now, as you can see in this diagram, that the start codon is AUG, whereas stop codon is UAA. Similarly, this is how these codons code for a specific amino acid, and hence mRNA helps in the synthesis of protein or the growing polypeptide chain. Now, as we have seen that there was triplet of the codons, what are they called as? They are called as genetic codons. This can be defined as the exact sequence of DNA nucleotide, which is read as 3-letter words or codons that determines the sequence of the amino acid in the protein synthesis. Okay? So the genetic code links group of nucleotide in an mRNA to amino acid in a protein. The letters which are present in the codons, that is A, G, T and C. These alphabets corresponds to nucleotides which are found in DNA, and they are organized into codons, and the collection of codons is called as genetic code. And for 20 amino acid, there should be 20 codons. Each codon have 3 nucleotides to impart a specificity to each of the amino acid for a specific codon. One nucleotide will have 4 combination and two nucleotide will have 16 combination and 3 nucleotide will have 64 combination, which is more suited for 20 amino acid which are present in our body. Now, there are properties of genetic code which are that these are read as triplet, that is the codon will be in the sequence of 3 or the combination of 3 nucleotide, that is it will be either UAA, UGE. Similarly, it is universal, like everywhere if UAA is in start codon, it will be a start codon. If it is coding for a particular amino acid, suppose the sequence AGT codes for phenylalanine, it is going to code phenylalanine. It is commaless and it does not overlap, like AGT, TGA. So the T is not going to be overlapped, it is going to be only in the sequence of 3 nucleotide. It is non-ambiguous, that is one codon will code only for one amino acid. It is degenerate and it has polarity. This was about the messenger RNA and the genetic code. Now we'll talk about the structure of tRNA. As we can look at this diagram, that it has a clover leaf-like structure. tRNA has clover leaf-like structure, as we can see in this diagram. It has 5 arms. So basically transfer RNA, it is soluble and it contains 71 to 80 nucleotides. The molecular weight of tRNA is approximately 25,000 and at least 20 species of tRNAs are present, which corresponds to 20 amino acids, and it is required for protein biosynthesis. It contains many unusual bases and nucleosides. The unusual bases which are present in tRNA are thymine and dihydrouracil. Hypoxanthine, I-methyladenine, and 2,9,2-N-methylguanine may also be present in tRNA. Now let us look at the structure of the tRNA. It has a clover leaf structure and it has 5 arms. Okay. One is your acceptor arm, second is your variable arm, third is your T-Cy-C arm, fourth is your anticodon arm, and fifth is your D arm. So these are the 5 arms which are present in the tRNA molecule. This diagram is very essential because it can be asked in the examination, the structure of tRNA, or that is when you'll have to draw this diagram along with the explanation. The CCA arm, that is cytosine adenine arm, it is present at the 3' end of the tRNA and it is an acceptor arm for the attachment of amino acid to form amino acyl tRNA. The D arm contains dihydrouracil. Third arm, that is T-Cy-C arm, contains pseudouridine. Anticodon arm contains sequence of 3 bases that are complementary to the codon mRNA. tRNA, it is also called as adapter tRNA because it carries specific amino acid on its 3' end because it हनी इफ इस पे लेजर नहीं आ रहा है. ये क्यों आ रहा है देखिए. यस. ठीक है, थैंक यू. T size C arm, it contains pseudouridine. There is anticodon arm which contains sequence of 3 bases which are complementary to codon mRNA. tRNA is also called as adapter tRNA because it carries specific amino acids on its 3' end corresponding to anticodon at its anticodon arm. The extra arm, it is also called as the variable arm, and based on the length of the variable arm, tRNA is classified into class 1 tRNA and class 2 tRNA. Class 1 tRNA contains short arm, that means it has 3 to 5 base pair, whereas long arm has 13 to 20 base pairs. tRNA accounts for 15 to 20% of the total cellular RNA. It is smaller in size and it is synthesized as precursor tRNA. Mature form of the tRNA is formed by post-transcriptional modifications. Now let us talk about the function of tRNA. As we have been discussing that this tRNA is involved in the protein biosynthesis. It has an anticodon arm to which the codons of the mRNA gets attached, and whereas in the 3' end, the amino acid complementary to this codon is synthesized. tRNA is required for protein biosynthesis. It is required for the transfer of specific amino acid to the site of protein biosynthesis. And it is also required for incorporation of a specific amino acid to the growing polypeptide chain. Now, what is a nucleotide analog? As the name suggests, analog means the structure is similar to the nucleotide and it is a synthetically synthesized molecule. Nucleotide analog is an organic chemical compound which is related to another by substitution of atom with other groups. So basically, there are analogs of a nucleotide in which there is change in the substituted group. They are prepared by altering the basis ring or sugar moiety. These are chemically synthesized nucleotide which are used as drug in the clinical therapy. So how it acts as nucleosynthesized analog. Since it is similar in structure, it is incorporated into DNA and RNA because of the structural similarity, which leads to synthesis of defective DNA and RNA, which inhibits the cell growth, which further inhibits cell proliferation, and hence used in the cancer treatment or as antiviral or in the treatment of gout by allopurinol drug. As we said that these nucleotide analog is used in the treatment of cancer, viral diseases, and gout. Okay, so now we'll talk about the nucleotide analog which is used to treat cancer. The pyrimidine analog, which are used to treat cancer is 5-fluorouracil, floxuridine, capecitabine, cytarabine, and gemcitabine. Now, the purine analog, which is used in the treatment of cancer, is 6-mercaptopurine, 6-thioguanine, azathioprine. It suppresses the immunological reaction during the transplantation. Mycophenolate mofetil, fludarabine phosphate, cladribine, clofarabine, and pentostatin. Antiviral drugs, or the analog which is used for the treatment of AIDS, is zidovudine and lamivudine. Now, these antiviral agent acts by interfering with the replication of virus by terminating the DNA synthesis. Zidovudine is also labeled as abbreviated as AZT, which stands for is azidothymidan, and it is used in the treatment of HIV. Lamivudine is used in the treatment of hepatitis B. The analog which is used in the treatment of viral diseases are zidovudine and lamivudine. These are sugar modified nucleotide analog. AZT is used in the treatment of AIDS, whereas lamivudine is used in the treatment of hepatitis. What it does, this basically interferes with the replication of viruses by terminating the DNA synthesis. Lamivudine, which is used in the treatment of hepatitis B. Now look at this diagram and how these purine and pyrimidine nucleotide analog acts as anti-cancer or antiviral, and at what level they inhibit the purine and pyrimidine metabolism. So pentostatin, it inhibits the enzyme adenosine deaminase, which is during the synthesis of ribonucleotide from the purines. This whole pathway will be looking in the purine metabolism. 6-mercaptopurine and 6-thioguanine inhibits the purine synthesis and also inhibits nucleotide conversions from ribonucleotide to deoxyribonucleotide. Methotrexate inhibits purine synthesis and inhibits DTMP synthesis. Cytarabine inhibits enzyme DNA polymerase and inhibits RNA function. Crisantaspase deaminites asparagine and inhibits protein synthesis. This was about the purine analog. Now we'll talk about the pyrimidine analog. So 5-fluorouracil inhibits DTMP synthesis. Bleomycin damages DNA and prevents the repair of DNA. Alkylating agents like mitomycin and cisplatin inhibits the cross-linking of the DNA. Camptothecins, doxorubicin, etoposide, amscarine inhibits topoisomerase 2 and inhibits RNA synthesis. Dactinomycin intercalates in the DNA, inhibits topoisomerase 2 and hence inhibits the RNA synthesis. Vinca alkaloids like taxanes inhibits the function of microtubules. Now look at the mechanism of action of azathioprine. Azathioprine gets converted to mercaptopurine. This mercaptopurine gets converted to 6-thiogTP. This 6-thiogTP inhibits RNA and DNA synthesis, and hence protein synthesis and cell division is inhibited. Now look at the mechanism of action of mycophenolate. What it does, it gets converted into mycophenolic acid, which inhibits IMP dehydrogenase enzyme, and hence the nucleic acid synthesis is inhibited. So this was about the mechanism of nucleosynthetic nucleotide analogs and their mechanism of action. So this was about the RNA and the nucleotide synthetic analogs. And during university examination, nucleotide analog is a very important question. So ensure that you write the nucleotide analog definition, the various types, and their mechanism of action along with the example. Thank you.